All right, so this is the first lecture of Unit 5, the genetics unit, and Chapter 32 is pretty jam-packed with content, so we split it into 2 lectures. The first lecture here we'll talk about principles of genetics, so we're going to go through a lot of definitions and terms and some basic concepts, so that in the subsequent lectures, when we go through the different genetic disorders, you can see how these concepts are applied to those clinical situations. So this is a diagram from Unit 1 where we talked about DNA structure and DNA replication. This is just a gene. And again, a gene is just a basic unit of inheritance. So the gene is within the DNA and has a specific sequence that codes for, obviously, RNA. And then RNA codes for protein through translation. So then you have a locus, which is the location of a gene on a chromosome. So here we have a diagram of a chromosome here. And this gene shown in red here, this has a specific location on the chromosome. Now, for a gene, genes can come in different versions or alleles. And so alleles are different variants of a gene. And you can have what's referred to as the dominant allele, which we show here in dark red. And then you can have the recessive allele, which is shown here in lighter red or pink color. And these are both the same gene. They're in the same locus on the chromosome, but they're just different versions of that. And so, again, this could be an example of eye color. So you could have brown eyes, or you could have blue eyes. Same gene, just different versions of it. Now, the genotype versus the phenotype, you hear these terms thrown around a lot. The genotype is the genetic code, the actual genetic sequence. So here, this would be the gene for eye color. And then the phenotype would be the actual physical manifestation of the gene. So say this person inherited the blue color allele, so they would code for blue eyes, which would be the phenotype. Then you can have homozygous, which is where both alleles of a gene are the same. And so you can have homozygous dominant or homozygous recessive. And so homozygous dominant would be, so see, you have the same gene here. It's in the same location or locus on the chromosome. You've inherited the dominant allele from both of your parents. Because remember, you inherit one chromosome from your dad, and then you inherit one chromosome from your mom. So your dad gave you the dominant allele and your mom, because you have 2 copies of each chromosome, 1 copy from your dad, 1 copy from your mom. In homozygous recessive, so both parents again, so your dad gave you the recessive allele for this particular gene, and then your mother gave you the recessive allele for this gene. Now in heterozygous is where both alleles are different. So for example, you can have your dad give you the dominant allele, and then your mother, you can inherit the recessive allele. So you have 1 copy of each, of the dominant allele and the recessive allele, and this is what's called heterozygous. Now obviously the reverse of this can happen. You can inherit the dominant allele from your mother, and you can inherit the recessive allele from your father. So mosaicism is the presence of 2 genetically differing cell lines in the same person. Now we're going to go into more detail about this in actually the Chapter 38 lecture, but just to give you a brief overview of what this actually means. So during development, we all start from 1 cell, and then the cell keeps dividing and dividing. And so what can happen is this is 1 cell line. But what can happen is you can have a mutation in one of these cells. And then that mutated cell divides, and so then you have essentially 2 populations of cells dividing. So this mutated population keeps dividing, while at the same time, the original cell population keeps dividing. So in the same embryo, you have 2 populations of cells that comprise the same person. And so this is what's an example of mosaicism. And this can result in a normal individual. It can also result in certain genetic disorders that we'll talk about later on. So then you have locus heterogeneity versus allelic heterogeneity. So let's talk about the difference of those. So locus heterogeneity is where you could have a mutation at one locus. So here you have a mutation X at this gene here shown in red, and it leads to disease X. But this can cause the same phenotype as a mutation from a totally different locus or a different location. So you have a mutation X here in this gene shown here in orange, and this also results in disease X. So you could have a normal version of the orange gene and a mutated version of the red gene and get disease X. On the flip side here, you can have a mutated version of the orange gene and a normal version of the red gene and still get disease X. This is an example of locus heterogeneity. Versus allelic heterogeneity is where multiple different mutations in the same locus can cause the same phenotype. So you have an X mutation here in this red gene, and that gives you disease X. And then you can have a star mutation here at the same gene, the same locus, and also get disease X. And this is an example of allelic heterogeneity. So anticipation, this is where you have earlier and more severe disease manifestations in subsequent generations. The best example of this is where you have trinucleotide repeats. And we have an entire lecture dedicated to trinucleotide repeats, what they are, and then the diseases that involve them. But just to give you a basic overview, so you have a sequence of 3 bases here, and it gets repeated. So in this example, it's CAG. So CAG keeps getting repeated in the genetic code, and it shifts the entire reading sequence, which, as you could imagine, can cause significant downstream effects. And so as you can see here in the first generation, you have this many repeats. And then in the next generation, you have additional repeats. And then in the third generation, you have even more repeats, as you can see here. And so with each generation, the disease is more serious and occurs earlier in life. And this is a very common example of anticipation. So variable expression is when you have the same genotype, but the manifestation of the phenotype may vary amongst different people. So you could have a number of individuals, they each have the same genotype, but you see different manifestations of certain genes among them. And those are due to epigenetic factors, which we talked about in Unit 1, where you have methylation or acetylation, things like that. You could also have just the environment. So if they're exposed to certain toxins or other certain factors that impact DNA or maybe would damage DNA, for example, if someone's a smoker or not, or things like that. And then they can also be affected by what's called modifier genes. And so these are genes that code for proteins that impact the expression of other genes. And so it depends on how these genes are expressed, are they mutated, things like that. This is a very complex concept, but we just wanted to kind of illustrate in the basic term what it is. Heteroplasmy is where you have the presence of both normal and mutated mitochondrial DNA. So remember, the mitochondria have their own DNA, and a person can inherit both normal and then also inherit some mutated DNA. So you can have some mitochondria that'll have the normal DNA. Some mitochondria could have the mutated mitochondrial DNA and coexist. And this can result in variable expressivity of mitochondrial disease. And so what happens as a result of this is that patients can manifest the disease in different variants because it just depends on what percentage of their mitochondria inherited this mutated mitochondrial DNA. Linkage disequilibrium is the chances of 2 different alleles at 2 loci occurring together are not random. And so let's just draw a chromosome here. And let's say you have gene A here, and then you have gene B here. And let's say you can have a positive allele or a negative allele, or this would be the dominant and recessive. And so you can have A plus allele and B plus allele. And so let's say you screen a population and you see a high occurrence of A plus allele being expressed along with the B plus allele. So you see a lot of individuals inheriting the plus allele at the A gene and the plus allele at the B gene. And you see them being expressed together. What this is saying is that the chances of that just being a random occurrence are in fact very unlikely, and so really these genes are what's called linked. And so you hear linkage disequilibrium is what this, in terms of genetics, is what this is called. And again, it's just stating that these genes, again, are very commonly found, expressing this particular version of the gene or allele of the gene, both at the A and the B gene in this case. Epistasis is where one gene affects the phenotypic expression of another gene. So an example of that in fungi is, let's say some fungi make penicillin, the antibiotic. And so let's say you have enzyme B, which is from gene B, and then you have the precursor, which would just be, we'll call that X. And then you have the synthesis of X, which is catalyzed by enzyme A. And so let's say you have perfect expression of gene B, but you have a mutation in gene A, and so you have deficient enzyme A. So that's an example of epistasis because the expression of A affects the phenotypic expression of B. So since even though you have a normal expression of B, normal functioning B, if you don't have this precursor, you can't make penicillin. And so as a result of this mutation in this gene, the product of this gene is affected, and thus the phenotypic expression of gene B is affected. So that's an example of epistasis. Codominance is when both alleles contribute to a phenotype. So a good example of codominance is blood type. So you can have IA and IB. These are both considered dominant. And then you can have the lowercase I allele. And so a person can have 2 copies of the IA, and this would be blood type A. They can have 2 copies of the IB, and this would be blood type B. And then they can have, so these would both be dominant alleles or dominant expression. And then they can have 1 IA and 1 B, and this was what gives a person the AB blood type. And so this is what's an example of codominance. And then obviously, lastly here, you can have 2 copies of the I, and this is O blood type. And then pleiotropy is where a single gene can have multiple phenotype presentations. And so an example of this is where if you have a mutation in a particular gene for a protein, say, that is involved in a lot of different processes or is expressed in a lot of different tissues, that can impact multiple different presentations. An example of that would be Marfan syndrome, which is where you have a mutation that affects connective tissue, specifically fibrillin and then elastin structure. And so that obviously is involved in a lot of different structures, and you can have variable presentation of that. You can have it where it just simply affects kind of the musculoskeletal system, and so you have different musculoskeletal presentation, and then it can also be more severe where it also involves cardiovascular problems as well. And so it just depends on that gene's impact on various tissues. And again, that just varies from individual to individual. So the concept of uniparental disomy, this is when both pairs of a chromosome are inherited from only one parent instead of both parents. So we'll come down here and take a look at this diagram here. This is actually from the lecture where we'll go more in depth on uniparental disomy and talk about some disorders that exhibit this. Normally you have one chromosome that's inherited from your father, and then another copy of that chromosome inherited from your mother. So let's say in this case it's chromosome 15. So you have your paternal chromosome 15 and your maternal chromosome 15. And this is what would be normal. But however, in uniparental disomy, you can inherit both copies from your father, which would be paternal uniparental disomy. So you didn't get any copies from your mother. So both of your 15s would be from your dad in this case. And then you can have maternal uniparental disomy, which is where you inherit both copies from your mother. And so you don't have any copies from your dad here. Incomplete penetrance. This is when an individual with a disease genotype does not actually manifest the disease phenotype. So penetrance is, in general terms, is the percentage of patients with a mutation that actually manifest the disease. Because just because you have the mutation does not always mean you'll actually develop the disease. So a real life example of this would be patients with the BRCA1 mutation. And this would be for breast cancer. And so what they've found is that actually 80% of patients with this mutation develop breast cancer. And so what that means is that this mutation actually has an 80% penetrance, because 80% of the patients with it end up going on to develop the disease. So 20% of patients with this mutation do not manifest the disease at all. And so that's an example of incomplete penetrance. Complete penetrance would be where 100% of people that have this mutation develop this disease. Consanguinity is when you have mating of genetically related individuals, and this often results in the increased likelihood of a genetic disease in the offspring. And examples of this in real life is when you have certain groups of people where you see higher rates of people mating that are genetically related. As a result of that, you often see a higher incidence of certain inherited genetic disorders, and that's what contributes to that partially. The concept of imprinting, so this is when a single allele of a gene is active instead of both alleles being active. And this is due to often methylation of the DNA resulting in inactivation of one of those alleles. So again, you inherit one chromosome from dad, one chromosome from mom. You have this gene at this locus or this location. We'll say this is chromosome 15 because that's in this diagram, that's what it's indicating here. And so what can happen here is you can have where the paternal gene is methylated, and so it's silenced, and then the maternal gene is expressed. And so it doesn't matter because for this gene to manifest its phenotype, you only need one copy being expressed. And so the other copy gets silenced, or the paternal. And then we have another example here where you have different gene, different location. You have one from dad, one from mom. And in this case, the maternal gene gets methylated and silenced or imprinted. And then the paternal gene is the one that's expressed. And so again, for this particular gene, you only need one allele or one copy being expressed to manifest a normal phenotype. And then here, what we have here, this is just a simple flow where you're exhibiting the central dogma of biology. We covered a lot of this in Unit 1, but again, it's important here for genetics. So you have DNA, which then gets transcribed to RNA, which then gets translated into the protein. And so this would be, expression of the gene in the form of the protein, and then obviously the function that the protein carries out. Again, this is the central dogma of biology. All right, so now we're going to talk about some terms relevant to chromosomes. So first we'll talk about karyotype. This is just a picture or a drawing of all 23 pairs of a chromosome next to each other. So, humans obviously have 23 pairs. Here's each of those pairs lined up together. This is useful for diagnosing chromosomal abnormalities such as Down syndrome, where you have trisomy 21. Talk about that in a second. And remember, you get 23 from mom, and you get 23 from dad, and that together gives you 46. So for each of these, one came from dad, one came from mom, and that's true throughout all of this. And so that's how you end up with 23 pairs to give you 46 total. So for chromosome structure, you can have a metacentric chromosome, which is where we'll draw this here. You have the centromere like this that's in the middle, and both arms are about equal to each other, as you can see here. So here's the centromere in the middle, and that would be metacentric there. And so the P and the Q arms have equal length. Then you can have what's called acrocentric, which is where the centromere is near the end of one of the arms. So we'll draw that here. And then usually the P arm is the shorter one, and then the Q arm is the longer arm, as you can see here. And this would describe the structure of chromosomes 13, 14, 15, 21, and 22. And these are the type of chromosomes that are involved in Robertsonian translocations. We'll talk in more detail about that in Chapter 35 lecture, where we talk about aneuploidies. So these terms refer to the number of chromosomes within a cell. So you have euploid, which is where a cell has a multiple of 23 chromosomes. So they could have 23, 46, 69, and so on. And so each of these terms describes what multiple of 23 they have. So first you have haploid, which are cells with one pair of 23 chromosomes. And this is what you would describe the sex gametes, so the sperm and the egg. So you've got 23 chromosomes in the sperm from dad, and then you have 23 chromosomes in the egg from mom. Then you have fertilization that occurs, and then you get 46 total, 23 pairs. Then you have diploid cells, which are cells with two pairs of 23 chromosomes. This is what would be described as somatic cells. So the cells that end up resulting from this fertilization are called somatic cells, which are essentially non-gamete cells. And so they will have two pairs of 23 chromosomes, one set from dad and one set from mom. And then you can have triploid, which are cells with three pairs of 23 chromosomes for a total of 69. And this is abnormal. This is usually when you have examples of non-disjunction, which is where you have unequal splitting of chromosomes during meiosis, and we'll talk a little bit more about that in a few slides. And then you can have aneuploid, which is where any number of chromosomes that is not euploid, so not a multiple of 23. This is often due to a loss of a chromosome, which would be monosomy, or a gain of chromosome, which would be trisomy. And so here, monosomy is, obviously a loss of a chromosome, and usually these do not result in live birth. So here's an example here where you're missing a chromosome 23. Trisomy, obviously again, is a gain of chromosome, and these may result in live birth when they occur with trisomy 13, 18, or 21. So you have an extra 13 chromosome, 18 chromosome, or 21. And here's an example of that here. So you have trisomy 21, where you have an extra 21 chromosome. And this is what is the underlying cause of Down syndrome. So we've mentioned this before, but the concept of non-disjunction is often the cause of aneuploidies due to a lack of unequal separation of chromosomes, either during meiosis 1 or during meiosis 2. The risk of non-disjunction increases with age, and so that's why mothers who are older, say in their late 30s or in their 40s, are at an increased risk of non-disjunction versus younger mothers who would be, say in their early 20s or mid-20s. And so this is a diagram that shows that. So during metaphase, you have the chromosomes lining up along the center of the cell. And then proper disjunction is where they're equally separated like this and like this. Now, non-disjunction is where you have unequal separation. So instead of both sides each getting one, both of them will go to one side. And again, this can happen during meiosis 1 or meiosis 2. All right, so let's go to the whiteboard here for a minute to show you the difference between non-disjunction during meiosis 1 and meiosis 2. So here you've got meiosis 1, and you have lining up of homologous chromosomes during metaphase 1. And remember, you would have duplicated the chromosomes during the S phase of the cell cycle. And so during metaphase 1, you have lining up of these homologous chromosomes. And let's say this would be for all 23, but let's just say here we'll show 20, 21, and 22. Now, during anaphase 1, you're going to be equally splitting up these homologous chromosomes in preparation for equally dividing them into the two daughter cells. And so let's just say you have normal distribution for 20 and 22, but let's say non-disjunction occurs for 21, where one of the cells will get both homologous chromosomes. So we'll have the daughter cells like this. And let's say for chromosome 20, each one gets a homologous chromosome of chromosome 20. However, this cell is going to get none for 21, and then this cell will get both chromosomes for 21. And then again, both cells will get a homologous chromosome for 22. So again, then you're going to have split of these during meiosis 2. And so each of the daughter cells here will get one sister chromatid from 20, because again, you're going to pull these apart. You're going to pull the sister chromatids apart. And so you'll get one for 20. You'll get none for 21. And then for 22, obviously you'll get one sister chromatid as well. So normal for 20 and 22, but abnormal here for 21. On this side, you're going to pull apart the 20 and the 22, and then both of these 21s. So what you'll have on this side for 20, 21, and 22 is each cell will get one copy of chromosome 20, and they'll have two copies of 21, and then one copy of 22. So let's go over here, and let's say again, we've got 20, 21, and 22. This is meiosis 1 here. You've replicated them in the S phase of the cell cycle. And let's say meiosis 1 goes off without a hitch. So here, you've got equal distribution of homologous chromosomes between the daughter cells. And again, remember, you're going to split these during anaphase equally. But let's say in this daughter cell, both of these chromatids will go to one side. So what will happen here, we'll draw a little divider line here, is you're going to get obviously equal distribution of 20 and 22. But then what will happen is one cell will get both of the 21s, and then the other cell will only get none. And then this cell will be normal, where you'll have one of each and one of each like that. So that's just the difference between meiosis 1 and meiosis 2 for non-disjunction. All right, so that closes out our discussion of the principles of genetics. And in the next lecture, we're going to talk about the different modes of inheritance and the Hardy-Weinberg equilibrium.